So let's begin discussing some of the specific features of our cells. In the last video, we talked about these four structures that are present in all prokaryotes and are actually present in all types of cells. So all cells have a plasma membrane, all have cytoplasm, DNA, and ribosomes. And we're going to talk about those, and we're going to talk about some other important structures as well. We're going to start by talking about the plasma membrane. So if you recall, the plasma membrane is this really thin green structure here. You can't even see it on this microscope image down here, but it's just a really thin, flexible layer. And again, present in all cells. Of course, there are some other structures outside of that plasma membrane on this eukaryote, and we'll talk about those in a little bit. Animal cells have them as well. Remember, animal cells are eukaryotic cells. And we're going to sort of talk about animal cells as kind of representative of all non-bacterial, non-plant cells, just really generically. So the plasma membrane would be this blue region on the outside of the cell. It's the outermost layer in an animal cell. And then if we were looking at a plant, it would be along the outside of the plant, but inside of the cell wall, sort of like that bacteria image over there. So the plasma membrane is made up of two specialized layers. And we'll talk about what actually makes up those layers in just a minute. Embedded within those layers are specific pores or channels. And those pores and channels allow things to move in and out. You can see there's a channel here, there's a pore here. This is possibly a pore or a channel. This is very likely another type of molecule. So there are also molecules that are attached to the outside, and those are usually involved in communication and recognition between cells. So the function of the plasma membrane is one, to sort of hold or contain the contents of the cell, and then also to regulate what leaves and enters the cell. So cells might need to take in food or nutrients. They might need to bring in other products within an organism from another part of the body. Say if you're a human, maybe the cell needs to take in hormones or proteins that are made somewhere else. And then each cell type, particularly within a eukaryotic organism, will also make things that need to be exported to other parts of the cell. So the plasma membrane sort of regulates all of this. It's sort of can be thought of as a gatekeeper or a border patrol. It really just regulates what moves in and out. And then, as I said, it can also help in interacting with the environment or neighboring cells, sort of sensing the environment around it. And once it can sense the environment, it can then make adjustments to its own internal environment if necessary. So the plasma membrane's really thin and it's really flexible. So cells that need to move have this flexible membrane outside of them. It doesn't really give enough support for, say, something like a plant where you need some sort of structural support. And so that's why we have in some of these bacteria and in plant cells, we have those strong cell walls, because the plasma membrane, although it does give a little bit of structure and holds the contents in place, it really isn't a strong molecule. It's real thin, real flexible. So the plasma membrane is made up of something called a phospholipid bilayer. Now, this is just a really fancy term for something that is basically a fat-like molecule, and a bilayer just means that there's two layers of it. Remember in the last slide I said that the plasma membrane is made up of two layers. So they are made up of two layers of these structures called phospholipids. And the structure of the phospholipid is really what gives the membrane its really important function of regulating what moves into and out of the cell. So let's look at the structure a little bit. There are two main components. There's the head of the phospholipid, and the head has a property called polar. So when a molecule is polar, it means that it is attracted to water, and so it will interact well with water. Now we call it a phospholipid, phospho meaning that there is a phosphorus molecule, a phosphorus containing molecule within this head. Now, we obviously didn't do the chemistry sections in our book, and that's okay. I don't expect you to know the chemistry behind this. I just want you to understand the properties. So the head of this molecule will interact with water. The tail of the molecule has a property called hydrophobic or non-polar. So hydrophobic molecules are those that are repelled by water, that do not interact with water. So non-polar means that it doesn't interact. Hydrophobic means basically it means water fearing. It is repelled by water. And this tail of the phospholipid is actually called a phospholipid tail. This tail is basically a fat, and we all know that fats, oils and fats, bacon grease, olive oil, things like that, don't interact well with water. So this phospholipid bilayer is really cool, and the way that it works is if you have phospholipids in a solution, they'll actually sort of self-assemble into this bilayer where you have the hydrophilic heads, the heads that are attracted to water on the outside and the inside, and the hydrophobic tails, the ones that are repelled by water on the inside. And you can see here that we form two layers, or a bilayer. So this bilayer structure is able to interact with the watery fluid outside of the cell and with the watery fluid on the inside of the cell. Now, those hydrophobic tails are actually what helps regulate what moves into and out of the cell. So other molecules cannot simply just move across the membrane. They are blocked by either they themselves being hydrophobic or hydrophilic. So if they're hydrophobic, they won't interact well with these heads, and if they're hydrophilic, they won't interact well with the tails. And so this membrane, just based on its structure, can keep other things from just moving through freely. And so that's why we call it a gatekeeper. We will start talking about how things actually move, and these pores and these channels are actually ways for molecules to move through the phospholipid bilayer. The phospholipids are not locked in place. They sort of just float around each other. So they can move laterally in a fluid manner, but they also can't pop out. This allows them to have embedded within them those channel proteins and pores, and it allows the phospholipid molecules to move around and maintain a fluidity and a flexible structure. But no matter what, they maintain this bilayer with the hydrophilic heads and the hydrophobic tails, keeping them in this structure. So the molecules that are embedded within the phospholipid bilayer are really important. There are a variety of different types of molecules, each with their own function. So here we have the phospholipid bilayer. You can see the blue hydrophilic heads and the kind of greenish yellow hydrophobic tails. Now, any molecule that's going to be embedded within this membrane has to have those hydrophilic and hydrophobic regions as well. So this transmembrane protein, that just means it's a large protein that stretches across the membrane. Trans means across. So this transmembrane protein has a hydrophilic region and hydrophobic region as well, and that allows it to remain sort of stuck within the plasma membrane. So we say that the plasma membrane is a fluid mosaic of proteins, lipids, and carbohydrates. Fluid meaning that it can move, mosaic meaning that it's made up of a bunch of different things. The proteins found in the plasma membrane enable it to carry out its gatekeeping functions. So the proteins within it are going to allow specific molecules or specific types of molecules into or out of that plasma membrane. Proteins can also act as receptors. They can act as sort of helper molecules to help other molecules sort of enter and leave the cell. And they can also catalyze reactions. That means help chemical reactions within the cell to go to completion. And those that do that can be inside or outside of the cell. So they can catalyze reactions on the inside or on the outside. There are specific proteins that are attached to carbohydrates. So we have one here in the middle. This is a carbohydrate. I'm sorry, this is a protein with a carbohydrate chain. So carbohydrate is a type of sugar, or a generic type of sugar. And those carbohydrate protein complexes are what helps identify one cell from another. So these are going to have be sort of like little beacons that identify what the cell is. We also have embedded in here a molecule called a lipid. Now, a lipid is a type of fat molecule. This one is actually a cholesterol molecule, which actually is a steroid. So there are a couple different types of lipids. Steroids are one type of lipid, and they're sort of related to fat molecules. Cholesterol is really important in maintaining the fluidity of the membrane. So I said a minute ago that the phospholipid bilayer was fluid and it could move. Well, if it warms up too much, it's kind of like melting butter. All of those molecules can sort of spread out. But cholesterol actually can block that. So it helps sort of stabilize the fluidity. And then also, if the membrane gets too cold, and so if you think of it the other direction, if you put olive oil in the refrigerator, it sort of solidifies. Cholesterol will actually keep the membrane fluid. So depending on the levels of cholesterol in the molecule, the cell can regulate the fluidity of the membrane, which is really kind of cool. So cholesterol is not all bad. You actually do need it for some very specific functions in your cells. All right, so let's talk about some of these specific molecules. We're just going to go left to right here. First, we have receptor proteins. So receptor proteins are going to bind to chemicals in the cell's external environment. So chemicals that are outside the cell. This might be a hormone. It might be some sort of nutrient that the cell needs. And in doing so, in binding those chemicals, they can actually help regulate certain processes within the cell. So if the cell needs a process to run, it can bring in those molecules. And if a cell doesn't, it can actually block bringing in those molecules. Then we've got those recognition proteins, those carbohydrate proteins. They sort of act as a fingerprint for the cells, or as I said on the last page, a beacon that identifies what the cell is. So these recognition proteins give each cell their own unique fingerprint. And this makes it possible for something like the body's immune system to distinguish between a cell that belongs in the body and an invading cell, or something like a virus. These recognition proteins can also help in allowing cells to adhere to one another or to a substrate, which is just something that a cell attaches to, depending on the situation. So those carbohydrate proteins are really important. The specific name for that is a glycoprotein. Glyco meaning glycogen, which is a sugar molecule or a carbohydrate. So it is a sugar molecule attached to a protein. Over here we've got another one of our, our recognition protein, a different type. This would be something that maybe the immune system cells identify and therefore don't attack the cell within the organism. So then we have transport proteins. Transport proteins are proteins that stretch across the membrane. So they are transmembrane proteins. And they help move large molecules that couldn't otherwise pass through the plasma membrane. And they also can help move strongly charged molecules to pass through the membrane. So remember we said that the plasma membrane had a hydrophilic side on the outside and the inside, and then a hydrophobic layer in the center. So molecules that have a charge, a positive or a negative charge, are actually hydrophilic. Water also has a slight charge to it, and therefore attracts other molecules that have a charge. So if you have something with a positive charge, it's going to attract a negative. And so because of that charge affinity, those molecules can interact on the outside or the inside, but they can't make it through. And so these transmembrane proteins are actually going to allow those large molecules or charged molecules, other types of polar molecules, across that non-polar hydrophobic region in the center. So finally, we have enzymatic proteins. So these are the ones that are going to help accelerate chemical reactions on the cell's surface. So there are a variety of different types of enzyme proteins. Some that help reactions on the inside, some help reactions on the outside. So just a quick recap then. We have the receptor proteins, the recognition proteins, the transport proteins, and the membrane proteins. So these are the four main types of proteins and the four main types of functions of the plasma membrane. And then, of course, we have our carbohydrates that act as sort of a fingerprint identifier for the cell, and then our cholesterol, which we discussed on the last slide, which helps to maintain and regulate membrane fluidity. So the single most common fatal inherited disease in the United States is cystic fibrosis. So cystic fibrosis is a disease that results from an improperly functioning membrane. So the plasma membrane, as we said, is really important in regulating what moves in and out of the cell. And cystic fibrosis is basically a defective receptor situated in, or multiple receptors situated in the cells that are not properly moving contents they're supposed to be. So we're going to do an assignment in class that discusses cystic fibrosis just a little bit. But basically, cystic fibrosis occurs when an individual inherits excuse me, two mutations from their parents for the cystic fibrosis chlorine transporter protein. So this the genetic information, this faulty genetic information, codes for a transport protein that allows chloride ions to get in and out of the cells. This transport protein is primarily in the membranes of cells along the digestive tract and along the lungs. So when individuals have cystic fibrosis, these chlorine ion transporters are not functioning correctly. And because of that, they get sort of a mucus that lines the insides of their lungs and other organs. So there are different types of treatments for this. One is sort of a thumping, like a mechanical thumping on the chest and back to sort of loosen this mucus. And then they also have these vests that deflate and inflate rapidly that help to relieve this mucus accumulation. So this is a major disease that all is related back to faulty membranes. Faulty membranes also play a role in many other diseases. Heart disease, which is basically caused by an inability to move cholesterol, diabetes, certain types of hormonal diseases. And in most of these cases, the membrane component that is improperly functioning is a receptor protein or a transport protein. So those receptor molecules are also Receptor proteins are also really important for cell membrane function. Remember, those receptor proteins help help cells identify critical incoming molecules. So a lot of pharmaceutical research focuses on altering cell membrane functioning. One group of drugs that alter membrane function are called beta blockers. These beta blockers are extremely effective at reducing anxiety symptoms. So the beta blockers work by binding to receptors in the plasma membrane that can also bind to adrenaline. So adrenaline is manufactured in a special gland called the adrenal gland, which is located just atop the kidneys here. So you are probably all familiar with adrenaline. This is that fight or flight response hormone. And it's important because it helps your body cope with stressful situations. So the receptors that bind adrenaline are called beta receptors. So in stressful situations, your adrenal glands pump out adrenaline. When reaching your heart or other locations in your body, the adrenaline binds those beta receptors, and then the adrenaline causes your heart to beat faster and more forcefully, increasing your blood pressure in the process. So this is the fight or flight. It's a short-term response where your body sort of pumps you up with blood pressure and gets your heart going so that you are ready to either fight or flight. So this is a short-term response, but it's not really healthy over the long run because if you continue with this fight or flight response, you can actually cause damage due to high blood pressure and damage to your blood vessels. So depending on severity, this reaction can also be problematic in just your day-to-day life. So if you're giving a presentation, you're taking a test, or if you're in some other anxiety-producing situation. So one of the pharmaceutical remedies for this is to take a beta blocker pill. So the beta blocker will dissolve, just like any other medication that you take, and travel throughout your body until they find beta receptors. So if you notice, they're actually similarly shaped to some extent to the adrenaline. So what happens is they actually bind those beta blockers in place of some of the adrenaline. And so you don't get such an extreme fight or flight response. So the outcome of this slows your heart rate. You don't get as much of the fight or flight response. This causes a reduction in blood pressure and can just basically relax the individual, calm down people who are having trouble with anxiety and things like that. So every cell in your body has sort of a fingerprint that helps identify the cell. So this is important so that your immune system can differentiate between your cells and something that's invading, a virus, a bacterial infection, because your immune system doesn't want to attack your own cells. So a fantastic example of this is the HIV virus and these special markers called CD4 markers. So cells in the human body have these CD4 markers, specifically on their immune system cells. So HIV can recognize these CD4 markers, and when the HIV virus recognizes the CD4 marker, it can bind to it and use that as a means to infect the cell. So an individual is exposed to the HIV virus. This is usually through body fluid contact. It's not through hugging, shaking hands, things like that, but you have to come in contact with infected body fluid. So the HIV virus will identify this CD4 marker, and when it does that, it knows that it's infecting the correct type of cell. So if the HIV virus is flowing through the system and it doesn't find if a cell doesn't have those CD4 markers, it can't infect them. So these cell markers can actually also cause problems as well. So if it's your immune system being attacked by a virus, obviously that's a problem, but your immune system will then be activated in response to the viral infection. But it can also be a problem. So all of our cells are identified with markers that are specific to us. So suppose you receive a liver transplant. Even if the donor is a close relative, the molecular fingerprint on their cells is going to be different than your own. So your body can attempt to fight that new organ because it identifies it as a foreign object. So during an organ transplant, doctors have to actually give specific medications to reduce the chances of your immune system attacking that organ. So they basically have to suppress your immune system until your body can tolerate that new organ, in this case, the liver. So this is important because it not only is reducing your immune system for bringing in that liver, but also your immune system as a whole.